Good evening to all the AP EAPCET champions. Yes, you are just one day close to the exam. Need not to worry. At this point of time, don't look into any new concepts. Just keep on revising and practicing. Revision also should be from your short notes beta. Whichever short notes or key notes that you have made and mnemonics that you have made, formulae that you have made, just go through all of that and most importantly look into all the previous year questions. And now in this short session we are going to practice all the questions, MCQs, which are most expected from the most expected domains of your chapters from botany, and previous year questions are also included. So yes, just for 30 minutes relax your mind freely and just focus on the class. Fine? And try to answer as many questions as possible and drop in the comment section. So now if we have to see the question from cell biology, like centrioles are dash structures that lie according to what? First, this question is about centrioles. You have to remember centrioles are non-membrane bound organelles you will see only in the animal cell. Quickly we will revise the concepts also beta. If you consider this as a cell, in that centrosome is present. Centrosome which is not bound by any membrane but just covered by a perinuclear space. Okay, and this is having two perpendicular centrioles in it. This is having how many centrioles beta? Two perpendicular centrioles which are just perpendicular to each other. So yes, centrioles are cylindrical structures which are perpendicular to each other, that lie perpendicular to each other. So yes, A is cylindrical and B is perpendicular to each other. Next, centrioles have an organization like what? Cartwheel organization, right? Wherein in centriole at the center you can see a proteinaceous hub. To that 9 radial spokes are present. To each radial spoke 3 peripheral microtubules are present like this, which are arranged in the form of a cartwheel as such. Okay, so this is arranged in the form of a cartwheel. Centrioles have an organization of cartwheels. Centrioles are made up of 9 evenly spaced peripheral fibrils of what? 9 evenly spaced peripheral fibrils which is made up of the protein tubulin, Each peripheral fibril of the centriole is a triplet. Okay, it is a triplet. And central part of the proximal region of the centriole is called as hub. This is proteinaceous hub. Not like the cilia and flagella, you will have 2 central microtubules here beta. Here you find one central proteinaceous hub. Okay. So here what you have to go through, this concept is all about centrioles. If you can analyze this concept and answer this question, you will not skip any question which is coming from the concept of centriole. So it is a cylindrical structure which is perpendicular to each other. Yeah, perpendicular to each other, and it has cartwheel organization made up of tubulin, protein and peripheral triplets are present. At the center it has proteinaceous hub. So here we are going with the option B. And next, if we have to look into the next question, this is based on the prokaryotic cell. Let us quickly remember and recall the prokaryotic cell as well. Prokaryotes are generally smaller and multiply more rapidly than eukaryotic cells. Yes, this statement is true. Glycocalyx could be a loose sheath. If it is loose, you are calling it as slime layer, while in others it is capsule. If the glycocalyx is thick, you are calling it as capsule. If it is loosely arranged, you are calling it as slime layer. And next, if you have to see, the plasma membrane of prokaryotes is structurally dissimilar to that of eukaryotes. No beta, the plasma membrane of prokaryotes is same as that of eukaryotes. There is no difference in that. That there is a difference with respect to archaebacteria, eubacteria, but there is no difference between prokaryotes and eukaryotes with respect to the plasma membrane. Next, chromatophores are the membranous extensions into the cytoplasm of cyanobacteria which contains pigments. True? Yes, chromatophores are the extensions. If this is the cyanobacteria structure, it has two types of cells. We will call it as vegetative cells and generative cell. It has plasma membrane extensions towards the cytoplasm. Here in cyanobacteria, we are not calling it as mesosomes, we are calling it as chromatophores. Chromatophores. It is because they are having colored pigments within it and they conduct the photosynthesis. They capture the sunlight and they conduct the photosynthesis. So here we have to go with the option. As he is asking you the incorrect statement, you have to go with the option C. Fine. And next, if you have to see, yes, here this question is about fimbriae and pillibuta. These are non-motile structures on the surface of the prokaryotic cell, both fimbriae and pilli. And here is the description. We have to analyze both assertion and reasoning. So here if you have to see, the fimbriae are elongated tubular structures made up of special protein. Wrong. What do we describe with respect to fimbriae? Fimbriae are bristle-like structures sprouting out of the cell. And pilli is the tubular extension which has special protein, that is pillis or pillin, that is the special protein of the pilli. So pilli is a tubular extension and fimbriae are the bristle-like structures that are sprouting out of the cell. So here if you have to consider both the assertion and reason, both are false beta. So here if you have to see, if you have to go for the correct answer, both the assertion and reason are false over here. And next, match column 1 and column 2, choose the correct combination from the given options. We have to choose the correct combination with respect to transport. This is from transport in plants. This is from transport in plants. So here if you see neutral solutes doesn't need any energy for their transport. They just transfer across the cells through simple diffusion. When it is through simple diffusion, what is the option we are choosing? Neutral solutes through passive transport. Next, water is transferred across the cells through the semi-permeable membrane from high concentration to low concentration of water transport, that is through osmosis. Next, polar molecules are requiring energy either through primary active transport or secondary active transport. So polar molecules is transferred through active transport. It requires the energy ATP. So here if you have to look into the options beta, A is 2 and B is 3, C will be option 1, option B will be the correct answer. Now there is one more question about the cell organelle. Okay, look into it carefully and try to answer. I am looking for your responses in the chat box. Good. Now read the given statements regarding a cell organelle. Okay, we have to look into the statements and we have to guess which cell organelle is that. It contains water, sap, excretory products and other unwanted materials. Of course that is true. It is bounded by single membrane, that is tonoplast. Just by looking into the single membrane tonoplast, you can go for the organelle, that is vacuole. But hold on, we have to look into the other three statements as well. In plant cells, it can occupy 90% of the cellular volume. True. Next, it contains cellular sap. Correct. It maintains the turgor pressure. That is also true. And here I would like to add on one more point with respect to vacuole. That is, all the excretory deposits from the plant cell are transported to vacuole. From the cellular constituents or cytoplasm through active transport. The deposition is through active transport. Fine. So this is one add-on point to it beta, just you can go through it. So with respect to all these features, which organelle we are going to choose, that is vacuole. Same you can get such type of questions describing you or asking you for the organelle, giving the characteristic features of that particular organelle. Now here we always get confused with the terms like cristae, cisternae and thylakoids, and now you should be clear with it. Flat membranous sacs in the stroma of the chloroplast. In the stroma of the chloroplast, flat membranous sacs, you are calling it as what? Thylakoids. These are thylakoids. Stalk of this sac-like structures is called as grana. Stalk of this sac-like structure is grana, and one grana is connected to other grana by what ma? Stroma lamellae. And next, if you have to see infoldings in mitochondria. Infoldings in mitochondria, that is, let us once quickly recall the structure of mitochondria. Mitochondria has two compartments, like first is, it is also double membrane bound organelle. It has outer mitochondrial membrane and inner mitochondrial membrane is infolded to cristae. Inner mitochondrial membrane is infolded to cristae, inner mitochondrial membrane. Fine. This inner mitochondrial membrane is making mitochondria into two compartments, right? Inner compartment is called as mitochondrial matrix and outer compartment is said to be intermembrane space. Do remember that. Now, this infoldings are called as what ma? Cristae. This is called as cristae. And next, disc shaped sacs in the Golgi apparatus. Sorry, here. This is cristae and disc shaped sacs in the Golgi bodies. You will call it as what? This disc shaped, where on one side it is cis phase or convex phase and on the other side it is trans phase or maturing phase. These are cisternae. So now it is easy to identify, right? First one will be option thylakoids, that is here, and next infoldings of mitochondria will be option 2 and this is option 3. So we have to go with the option C, right? Now next question. Yes, here both column 1 and column 2 are given. We have to choose on with respect to the organelle and its function. Dictyosomes. Dictyosomes are nothing but single unicisternae is present in plant cells. Just single cisternae is present in plant cells. You will call it as dictyosomes. So dictyosomes, of course, Golgi bodies, it is unicisternae or multicisternae. Its function is secretion and packing and forming glycolipids and glycoproteins. So here dictyosomes function is what? Secretion. Next, mitochondria. It is the powerhouse of the cell because it conducts cellular respiration or the synthesis of ATP. So here mitochondria is related to aerobic respiration. We have to go with option 5. Vacuoles, just now we have seen this is storing all the excretory product sap inside the plant cell. Next, grana. This is the main site for capturing of the light for photosynthesis. All light dependent reactions happen in grana. So here this is related to photosynthesis. So now what will be the correct answer, ma? Just type on and say me. What is the correct answer here? A is 4, B will be 5, C is 1 and D is 2. Option A will be the correct answer. Very good. Now here we have to read on these statements. This is about centromere. Of course we can relate this centromere, and this is about shapes of the chromosome. We have 4 different shapes of the chromosome, and here I'd like to keep you one question. In which phase of cell division you can clearly view the shapes of the chromosome? In which phase of cell division you can clearly see the shapes of the chromosome? In which phase, ma? Anaphase of cell division, right? In which phase you can see? Anaphase of cell division. You can clearly see the shapes of the chromosome. One more question for you. In which phase can you see the clear morphology or structure of the chromosome? clear morphology or structure of the chromosome is clearly seen in the metaphase. Fine. Now, centromere is present in the middle of the chromosome forming two equal arms. Then what is that shape of the chromosome? Metacentric. It forms V-shaped chromosome. It forms what shape beta? V shape. Now, chromosomes has a terminal centromere. It has terminal centromere means it is forming telocentric chromosome, right? It has only one arm of the chromosome. Next, centromere lies close to the end of the chromosome forming one extremely short and one extremely long arm. This is acrocentric chromosome. One extremely short and one extremely long, that is acrocentric chromosome. Next, centromere lies slightly away from the middle of the chromosome resulting into one short and one long arm. One short and one long arm. This is what, Nana? Submetacentric chromosome. So here we have to go with the correct options. First will be metacentric chromosome, second is telocentric chromosome, and third is acrocentric, fourth will be submetacentric. So here we have to go with the option A. Next question, ma. See, this is your previous year asked EAPCET question that is also from AP EAPCET. And this question is about nucleosome. Okay. Once let us quickly recall what is a nucleosome model. In nucleosome model we have learned the packaging of DNA, right? The negatively charged DNA is wrapped around the positively charged histone molecules. DNA which is negatively charged is wrapped around the positively charged histone octamers. So histone octamers has 4 histone molecules. What are they? H2A, H2B, H3 and H4. These are the 4 histone molecules which occur in 2 pairs. They occur in 2 pairs. 2 pairs of this 4 histone molecules are present and they are composed with what amino acids? Answer to this question beta, they are composed of what amino acids? Lysine and arginine, right? Basic amino acids. That gives it positive charge. Positive charge. So as such it forms one nucleosome. Like that, one pair of this is one DNA is attached to another by a linker DNA. Here it has H1 histone proteins. This all together it forms one nucleosome. So here the structure, you can clearly see, ma, the structures that appear as beads on string in chromatin and number of base pairs are in the nucleosome. Beads on string, it is because of the, what is that model? That is nucleosome, and one nucleosome is having how many base pairs? 200 base pairs. It is having 200 base pairs. Very good. Now next. Yes, this is also previous year asked AP EAPCET question. A non-staining secondary constriction at constant location of few chromosomes is called as non-staining secondary constriction. Now here we have to look into the chromosome structure. Just look at it properly. So a chromosome here has how many arms, ma? Has how many arms, and what is the primary constriction and secondary constriction here? Just tell me. Okay. This you will call it as primary constriction, fine. And how many arms how many chromatids does it have? 2 chromatids, 2 chromatids. This is chromatid 1 and this is 2. Each chromatid has 2 arms. And where is the secondary constriction here? This is the secondary constriction. You will call it as satellite DNA. You will call it as satellite DNA. Hello Gautam. So this is satellite DNA. Secondary constriction here is satellite DNA. It has more of repetitive DNA sequences. Repetitive, just a second, DNA sequences. So yes, here if we have to go with the secondary constriction at constant location in the chromosome, that is satellite DNA. Next question. dihydroxyacetone 3-phosphate and glyceraldehyde 3-phosphate are interconvertible. When you are looking into these two substrates, you have to remember the chain, that is glycolysis. Glycolysis, which is the first biochemical pathway or the first reaction that is operated both in aerobic respiration as well as anaerobic respiration. Glycolysis is common. So we know that glycolysis is also called as EMP pathway, and it is a sequential step of 10 biochemical reactions. How many biochemical reactions beta? 10 biochemical reactions, of which first 5 you will call it as preparatory phase, next 5 is payoff phase. So at 5th step, if you can see, 6 carbon is isolated to how many carbons? 3 carbon and 3 carbon. 3 carbon dihydroxyacetone phosphate and 3 carbon glyceraldehyde 3-phosphate. They are interchangeable through isomerases. They are interchangeable through what? Enzymes? Isomerases. So here is the question about DHAP, dihydroxyacetone phosphate, and glyceraldehyde 3-phosphate are interconvertible. The enzyme responsible for the interconversion belongs to the category of isomerases. That is option A. Next, which two groups of the following formula are involved in the peptide linkage between different amino acids? We all know 2 amino acids are linked to each other by the N terminal of one amino acid is linked to C terminal of the preceding amino acid. So N terminal and C terminal, they are recorded as, they are connected as such. 2 amino acids are connected by N terminal and C terminal. So here if you have to see the amino acids, how are they named, Itma? The 4 groups of the amino acid. Amino group is named is marked as 1 and hydrogen as 2. R group, that is a functional group, is named as 4 and carboxyl group as 3. So here in this question he is asking us peptide linkage is formed at what positions? It is in between 1, that is amino group of one amino acid, and carboxyl group of the other amino acid, that is 1 and 3. Option C will be the correct answer. Next, so Gautam, what is mean noble gas? Noble gases are inert gases. Chemistry sir will be telling you it in more detail. You can join sir's live session and ask more about chemistry questions. Yes. So read the given statements. Fructose is the sweetest sugar. So this is from biomolecules again. Fructose is the sweetest sugar, correct. Glycine is the simplest amino acid because R group here is just hydrogen. Next, lactose is a disaccharide composed of one molecule of glucose and the other molecule of galactose. True. Next, cellulose is unbranched chain of glucose molecules linked by 1,4-glycosidic bond. Of course, that is true. That is linked by 1,4-glycosidic bond. So here all the 4 statements are correct, right? So they asked us to choose the correct statement. So all the 4 statements are true. Next question. Yes. Again, here you can see 2 important questions from cell division. First, before answering this question, let's quickly recall, before answering this question, just quickly recall the cell cycle. So in cell cycle, if you have to see, in cell cycle, you have what? 2 phases, right? Interphase and M phase. Interphase has 3 subphases, G1 phase, S phase and G2 phase. And here divisional phase, that is M phase. Like that you have on total 4 phases. So here if you have to see what will be the fate of chromosome number here. Chromosome number in S phase, DNA duplication takes place, but there is no chromosomal duplication. Keep that in your mind. There is no chromosomal duplication, only DNA duplication takes place in the F phase. If M phase is mitosis, there is no chromosomal reduction here. Okay, there is a chromosome doesn't get half in the mitosis, chromosome number remains same. What if it is meiosis? What if it is meiosis? Meiosis, chromosome number gets half. Now keep this point in your mind and look into this question. How many chromosomes will the cell have in G1 phase, S phase and after M phase respectively if it has 14 chromosomes at interphase? If it has 14 chromosomes in G1 phase, of course it has 14 chromosomes in S phase also, right? Yes, and if it is meiosis, then it gets half to 7 at the end of meiosis. But here he didn't mention M phase whether it is mitosis or meiosis. So we are considering it as mitosis and chromosomal number remains unchanged. Now what option we have to go through beta? Number of chromosome number will be same in G1 phase, S phase and after the M phase also chromosome number remains same. That is option B. If in case here instead of M phase he is clearly stating meiosis, then what option you have to choose? 14, 14 and at the end of meiosis chromosome number gets half. So you have to go with the option C if it is asked with respect to meiosis. Next, identify the given stages of mitosis and select the correct answer. Okay, they asked us with respect to different stages of mitosis. Here you can clearly see the chromosomes are splitted to two separate spindle fibers or two asters. This is clearly stating you which phase? Anaphase. And here you can see all the chromosomes are in the equatorial plate. This is clearly giving out you metaphase. And here you can see thread-like structures and a spireme stage. Spireme stage here is what? Reflecting prophase. Next, this is the last phase wherein two different cells are formed, two daughter cells are formed. This is indicating telophase. So now you have to go with the correct option, ma. First A is what? Anaphase. A is anaphase. Next B will be metaphase, C is prophase and T is telophase. So here we are going with the option C. Now, assertion reason type of question and one more logical question from cell division and cell cycle. During anaphase, the centrosome of each chromosome splits and chromatids separate. Yes, assertion is true. When reason is given like during anaphase, chromatids move to opposite poles. Exactly true. Chromatids move to opposite poles. So here both assertion and reason are true and they are separating and moving to poles. But here there is no way in the reason, here there is an explanation given about spindle fibers. And here comes the doubt for you. Whenever option A, whenever in the assertion reason question beta, both assertion is true and reason is true. Here comes the dilemma for us, whether we have to go with the correct reason explaining assertion or reason not explaining assertion. At that time, you have to read the reason twice and thrice along with the assertion, putting the questions like why, what, because, if, like that you keep after assertion and read on the reason statements 3 to 4 times, then it will be clear for you on what you have to choose. So here if there is an explanation given about spindle fibers or asters on how the chromatids are moving to opposite poles, then you have to go and choose that reason is explaining assertion. Here just he gave a statement that chromatids move to opposite poles, so we are not going to choose that. Here we have to go with if both assertion and reason are true, but reason is not explaining assertion. Reason is not explaining why chromatids are separating and moving to the opposite poles. So here we have to go with the option B. Next one. What do 1 and 2 represent in the flow chart of meiosis 1 and meiosis 2? In meiosis 1, at metaphase 1 what is formed beta? At metaphase 1, bimetaphasic plate is formed. Bimetaphasic plate is formed at metaphase 1. In anaphase 1, there is no centromere splitting, but this part of the homologous chromosome moves to this pole and this moves towards the opposite pole. There is no centromere splitting, and here from the transition of metaphase 1 to anaphase 1, chromosome number is getting half. Understood? Here you can see 2 chromosomes, but here if you have to see, 2 chromosomes are separated to the opposite poles. They are separated to the opposite poles, indicating only 1 chromosome per cell. So here after meiosis 1 in these 2 daughter cells, what you can see, half the set of chromosomes, right? Here you have complete set of chromosomes in metaphase 1. In anaphase 1 it is getting half, means 2 daughter cells after telophase 1 are having half the set of chromosomes, so it is haploid. From here to here 4 daughter cells, this is also haploid. So here after meiosis 1 it is haploid, after meiosis 2 also it is haploid, so we have to go with the option B. Now here this question is based on the substages. This question is based on the substages of cell division. Clearly focus on this. So here you have to see pachytene. Pachytene means where crossing over is happening or recombination. This is an enzyme mediated process. What is the enzyme involved here, beta? Recombinase. Fine. So here D will be option 1. Next, zygotine. Whenever you get the terms like zygotine is the second substage of which phase, beta? Prophase 1. Whenever you get the terms like homologous chromosomes, bivalent, tetrad, these statements you see like synapses, synaptonemal complex, all this are related to zygotine, that is the second substage of prophase 1. So here pairing of homologous chromosomes, that is formation of synaptonemal complex, this is related to option 2. Next, anaphase. Centromere splits and chromatids will move apart. So this is option 3. Next, metaphase. Chromosomes move to spindle equator forming an equatorial plate. This will be option 4. Now you tell me which is the correct option. You tell me which is the correct option, ma? Yes, very good, Lavanya. It is option 3. Fine. Next. Engelmann observed that motile aerobic bacteria, this is from photosynthesis in higher plants, focus on it, and this is from the experimental part. Listen and choose carefully. Engelmann observed that motile aerobic bacteria accumulated near the areas of a filament of cladophora. This is accumulated near the filament of cladophora which were illuminated by red and blue light. Accumulate near the filament illuminated with green color, where it is not accumulated when it is exposed with green light from the infrared. That means we have to choose what statement. Once let us quickly recall the Engelmann's experiment. Engelmann has done what experiment, beta? Oxygen is released from water in the reaction. No, Subhashita, it is not option C, ma. Once look into the question properly, see, it is stating that, so here, yeah. So yes, it is stating that first a light is exposed onto a prism. Once let's recall his experiment. Light is exposed to a prism. After light exposing to prism, we all know the light will be scattered into VIBGYOR. The light will be scattered into VIBGYOR. Okay, like that, this VIBGYOR light is exposed to a filamentous algae. This is exposed to a filamentous algae. What is that filamentous algae? Cladophora. Over this filamentous algae, bacteria is placed. Over this filamentous algae, what is placed? Bacteria is placed, and that is aerobic bacteria. Aerobic bacteria. We all know aerobic bacteria at the end of respiration, it will evolve what? Oxygen, right? So it will it requires oxygen for the respiration. Aerobic bacteria requires oxygen for respiration. It will accumulate wherever there is more synthesis of oxygen. and cladophora at the end of photosynthesis it will evolve oxygen. As such, in the regions of blue and red light, after some time he has observed two peaks. He has observed two peaks, one peak at blue light, one peak at red light, indicating that maximum oxygen is evolved at the blue light and red light regions, and here aerobic bacteria got accumulated. And with this experiment he has given both action spectra and absorption spectra, and he said that red and blue light are more effective in photosynthesis. And Subhashita, option C, if you see ma, oxygen is released from water in light reaction. This is said by Jan Ingenhouse. Okay, no problem, learn it from here ma. This is from Jan Ingenhouse, who conducted experiment on hydrilla plant. Okay beta. So yes, oxygen is released from carbon dioxide in light reaction. This is nowhere explained. Chlorophyll absorbs green light, that is from the photosynthesis equation. So yes, here we have to go with option D. I hope this explanation is clear for each and every one ma. Is that fine? Yes, good. Now, reduction of NADPH in the chloroplast takes place during, reduction of NADPH takes place during what? Non-cyclic photophosphorylation that happens in higher plants, oxygenic photosynthesis. So yes, production of NADPH in chloroplast takes place in non-cyclic photophosphorylation at the end of what? Z scheme, at the end of Z scheme. Fine. At anywhere, anything if you want me to explain, just ask me ma. Any concept need to be dealt, you can ask me. At this point of your preparation, anything you want to revise as such, also you can ask me. Now, recognize the figure and find out the correct matching. So here the chloroplast structure is given and we have to go with the proper labeling. That is very simple, right? But then we make mistakes sometimes, just not looking into it properly. If you see clearly here, B is what? Stroma lamellae. B is what? Stroma lamellae. Where is that? So here this you can go. B is stroma lamellae. Everywhere it is given same. Next, A is what? Grana or thylakoid membrane. Both you can choose. A is grana. Next, these small structures are what? Ribosomes. And what type of ribosome you can see in chloroplasma? 70S ribosomes. 70S ribosomes. Next, this is large starch granules where the glucose is stored inside the chloroplast in the form of starch granules. And these are what? Lipid droplets. Lipid droplets. As such, if we have to go with the options, A is grana, A is grana, B is stroma lamellae, D is ribosomes, D is lipid droplet. So yes, D is lipid droplet, E will be ribosomes, and C is starch granule. So we can go with option A. Fine, understood everyone? Next. Yes. So here again, during the fixation of one molecule of carbon dioxide in C3 cycle, number of ATP and NADPH required are, okay, for the fixation of one carbon dioxide. In C3 cycle, what are the three steps that are happening? Carboxylation, reduction and regeneration. Three subsequent steps, what are they ma? Carboxylation. After carboxylation, carbon dioxide is fixed into two molecules of 3-phosphoglyceric acid. 3-phosphoglyceric acid, how many? Two molecules of 3-phosphoglyceric acid are fixed. And during the time of reduction, these 3 molecules of phosphoglyceric acid are turned to 2 molecules of glyceraldehyde 3-phosphate, and here it requires 2 molecules of ATP and 2 molecules of NADPH. Fine. And this 2 molecules of G3P, of which 1 carbon will be escaped for glucose molecule formation. Rest 5 carbons are regenerated, and at the time of regeneration 1 more ATP is used. As such, for 1 carbon dioxide fixation, it requires 3 ATP and 2 NADPH, which is option A will be the correct answer. Next, law of limiting factors. Whenever law of limiting factors comes in your question or to your mind, the scientist who has given law of limiting factors is Blackman. What does this say? This states that any biochemical process, if it is affected by more than one factors, then its rate will be determined by the factor which is nearest to the minimal value. So here we have to go with the option A, that is Blackman. Next. So here next, glycolysis is not needed when glycolysis is not needed. Here you have to remember, very good, Veronica. Is it option A? No. Once you recall the amphibolic pathway, ma? Amphibolic pathway. So in amphibolic pathway, in that chart of our respiration, we can remember that out of 3 respiratory substrates, that is carbohydrates, proteins and lipids. Fine, out of 3 respiratory substrates, carbohydrates, of course they have to go with the chain of glycolysis, Krebs cycle and ETS, that will be the chain. But protein molecules will join directly to pyruvic acid, that doesn't need glycolysis. How? Protein molecules, after breaking into amino acids, directly in this biosynthetic pathway, they can join the respiratory pathway through pyruvic acid. As such, lipids or fats also. Lipids or fats, you can consider it as fats better. It is break down to fatty acids and glycerol. Fatty acids and glycerol, wherein fatty acids can join the cycle through acetyl-CoA. Acetyl-CoA. Keep that in your mind. So it is both. Okay, good, Veronica is the answer for the previous question. Good, ma. Next. So here fats breaking down to fatty acids and glycerol. Fatty acids will join the cycle through acetyl-CoA, and proteins through amino acids, they will join the cycle through pyruvic acid. So both of them doesn't require glycolysis to join the respiratory pathway. And here there is more than one option that is correct, both for fats and proteins. So we have to go with the option D. Next, the next question is about tricarboxylic acid cycle. The formation of citrate is followed by 2 successive steps of decarboxylation. What are the products formed after each decarboxylation step? So here, after isocitrate, okay, formation of isocitrate, after isocitrate what happens in the cycle? Just recall immediately. Isocitrate, which is 6 carbon compound, is decarboxylated to 5 carbon alpha ketoglutaric acid. Alpha ketoglutaric acid, wherein 1 carbon dioxide is eliminated, and here there will be a release of NADH. Fine. Now, alpha ketoglutaric acid is converted to 4 carbon succinyl-CoA. Succinyl-CoA, wherein this is 4 carbon and 1 carbon dioxide is eliminated, and here also NADH is released. So with this you can clearly say the step 1 after isocitrate will be alpha ketoglutarate, and then it is succinyl-CoA. So it is option A. Done? Next. Link between glycolysis, Krebs cycle and oxidation of fatty acids or carbohydrates and fat metabolism. This is with respect to what is the linking molecule between glycolysis and Krebs cycle? Acetyl-CoA, and acetyl-CoA is also in the biosynthetic pathways of carbohydrate and fat metabolism. Remember that beta, it is acetyl-CoA. Next, here comes the tricky question, that is total number of ATP molecules produced per glucose molecule in eukaryotic cell. Fine. So here whenever eukaryotic cell is mentioned, we have to go with the option 36 total ATP are produced. If it is not given specifically in the eukaryotic cell, just they are asking you in aerobic respiration, then you have to go with 38. Why there is reduction of 2 ATP in the eukaryotic cell? Let's quickly recall the energy sheet of aerobic respiration. Of course, 4 consequent steps glycolysis. At the end of glycolysis or in the process of glycolysis, 2 ATP, that is 2 net ATP are formed and 2 NADH are formed, right? 2 NADH. 1 NADH is equal to 3 ATP, right? So 2 NADH, 2 into 3, that is on total it is 6 ATP. On total it is 6 ATP, which is from 2 NADH. Clear? Next. After glycolysis it is link reaction. Link reaction, 2 link reactions will happen for 1 glucose molecule. So here how many? 2 NADH again, 2 NADH, which gives out to be 6 ATP. Next, that is TCA cycle, Krebs cycle. Here also 2 TCA cycles will happen for 1 glucose molecule. So what will be the energy sheet here? Here on total 6 NADH, 2 FADH2 and 2 ATP are released out. 2 NADH2 means 6 NADH2 means how much 6 into 3, 18 ATP, and 1 FADH2 is equal to 2 ATP. In that case 2 into 2 is 4 ATP. Next, and it is 2 ATP. If you calculate the complete, it will be 38 ATP on total. But here what happens is, if you consider in cytoplasm, glycolysis will take place, where glucose is partially oxidized to 2 molecules of pyruvic acid. This pyruvic acid directly enters to mitochondrial matrix. This will directly enter into mitochondrial matrix. ATP molecules are utilized over here, but this 2 NADH to pump into this over inner mitochondrial membrane, there is a shuttle system activated called as glyceraldehyde 3-phosphate shuttle system. Okay, here what shuttle system is there in inner mitochondrial membrane? Glyceraldehyde 3-phosphate shuttle system. This shuttle system will utilize 1 ATP to transfer 1 NADH molecule. As such, to transfer 2 NADH molecules, it utilizes 2 ATP. This shuttle system utilizes 2 ATP. That is why if it is asked in the eukaryotic cell, we have to choose 36 ATP on total. Need not to confuse Just to pump the NADH from cytoplasm to the mitochondrial matrix for 1 NADH, inner mitochondrial membrane has a shuttle system, glyceraldehyde 3-phosphate shuttle system. It utilizes 1 ATP for 1 NADH to be transported. That's it. Keep it in your mind. Next. Yes. Here is a question on ETS, and just one more question. 2 questions beta. Hold on and just practice. We can definitely expect a question from electron transport system for sure. Okay, hold on for just 5 minutes again. So here if you see we have to, he has given us the locations and labelings we have to choose. Of course, inner mitochondrial membrane, protons will be pumped from where to where Protons are pumped from mitochondrial matrix to intermembrane space, right? Protons are pumped from mitochondrial matrix to intermembrane space. And here if you have to see, second labeling is with respect to, so A will be mitochondrial matrix, B will be intermembrane space. And this complex, that is complex 3, which is in between 1 and 2. Complex 3, it is cytochrome BC complex. Complex 3 is what? Cytochrome BC complex. And this is cytochrome C. Fine, mobile electron carrier, that is cytochrome C. Next, E is also cytochrome C and F will be cytochrome A. This is complex 4. Fine. Now, if we have to choose the options, A will be mitochondrial matrix, B is intermembrane space, and C is cytochrome B, D is cytochrome C1, and E is cytochrome C. F will be cytochrome A, and last it is cytochrome A3. Fine. So B will be your correct answer. Just we have to label them. Okay. Immediately, like proton pumping is from mitochondrial matrix to intermembrane space, and then you have to go for complex 3 mobile electron carrier, that is cytochrome C, and last cytochrome oxidase complex, that is complex 4, where you have cytochrome A and A3. That's it, simple. Next, last question, just looking into diagram, tell me whether it is epicotyle seed germination or hypocotyle seed germination. Tell me what type of seed germination it has, beta. So yes, Lucky, for 1 NADH it is accurately equal to 2.5 ATP ma, and for 1 FADH2 it is 1 ATP, it is 1.5 ATP. But for approximate calculation, for 1 NADH we will go for 3 ATP, and for 1 FADH2 we will go for 2 ATP. That is approximate calculation. Accurately it is 2.5 and 1.5 beta. Next. Here we have to recognize and find the correct labeling. First you tell me this is hypocotyle seed germination or epicotyle. Very good ma, that is hypocotyle seed germination. How? See if you can see here clearly. First, from the seed, if hypocotyle is germinating down, and if you can see cotyledons above the ground, that is hypocotyle seed germination. So here this C is what? Hypocotyle. So where is that? C is hypocotyle, and A are cotyledons. A will be cotyledons, and B will be epicotyle here. B will be epicotyle. The fundamental difference between epicotyle and hypocotyle seed germination, wherein epicotyle seed germination, epicotyle will come first and cotyledons will be never above the ground. Cotyledons are buried inside the ground itself. In hypocotyle seed germination, cotyledons are out above the ground and first hypocotyle will originate. So with this we have the complete practice session of all the chapters. Just missed out is sexual reproduction of flowering plants and genetics. I have already made many PYQ session on genetics. You can view that in the video section of NEET World. Just go through that, and I would like to say a very all the best and perform best. Good luck for all the AP EAPCET aspirants. You will definitely do well. Be confident and cool, and be confident, extremely confident. Don't even have a single doubtful thought of yes, how would be the question paper and what will be the competition, where I will be going and what college I will be entering to. These thoughts and all don't keep in your mind. Just be confident and be cool, calm and composed, and be positive that you are going to rock the exam for sure. All the very best ma. Thank you.